but we'll see what happens. Okay, so we got a quiz on Friday. There's a help session on Thursday at 8:30 p.m. on Google Meet. The link's in the syllabus. Monday and Wednesday, I do appointments 1 to 5 p.m. And you can just make those in class the day of. So if you want to talk to me today, great. I have somebody, I was Jen Kemmer coming in at 3:45, but even if that was the only time you could make, we could probably work something out. Let's see. So those are the things we've got going on in terms of lecture stuff. We have lab tomorrow. There will be a pre-lab. You have it in your hand, and we're going to work on it in class today. Okay? So you may get a lot of it done, or you'll know how to do it. Hopefully everyone will know how to do it by the time we get out and won't take much time. Pre-lab due in your hands. So pre-lab's due at the beginning of lab. Okay, your lab report's due by tomorrow at midnight. Lots of stuff going on here. No wonder I'm so busy. Lab due online. It's on Turnitin by 11:59 tomorrow. I think that's it. Did I miss anything? Okay. Sophia, you had a question about the lab report. Do you want to? So let's go ahead and get the lab report directions off the website. And then it was down here. The experimental part right there. Okay. Yeah. Yeah, you should include it because the RF values, what you scraped off, is a method thing, right? Tells you which ones, and in data you're going to use it to describe stuff, if that makes sense. So for me, that's something you did. I scraped it off at this RF value, and then you need the data actually later. So the RF values, you don't analyze it in the experimental, right? And in fact, what do? We have a data, and then we have a results and conclusions. You don't discuss it in data either. Your just data is your data, and then this is where you're going to use it to explain your results. Does that make sense? Okay. Not that I'm really, if you put it in the wrong section, I don't know that I'm going to take any points off. I might miss it, and you might have to come back because I'd be looking for it in one section and not the other. That doesn't mean you can't come back and ask for points back on something if I missed it. You certainly should do that. Okay? Other questions? All right. So we're going to talk about things you do with DNA today. We're going to talk a lot about plasmids. Tomorrow's lab is going to be, you're going to have a plasmid unknown, and you're going to make a map out of it. And there's some, before we do that, there's some concepts we need to talk about. Okay, so first off, a plasmid. Plasmid is a relatively small circular piece of DNA. And when I say relatively small, when we talk about like a unit of base pairs, right? We have a base pair, and then another base pair, and another base pair in DNA. That would be, you know, less than, wait, sorry, I'm trying to figure out. Yeah, less than, say, 20,000 base pairs. Generally, that's about how big they are. Circular piece of DNA. That means the ends are linked. Okay, which makes another thing important, concept important. We're going to talk a lot about plasmids, but another concept is that DNA can supercoil. Okay, that means it overwinds or underwinds. Either of those. Essentially, if you take a piece of DNA and you overwind it, it undergoes stress and it curls all around in funny ways. Okay? Now, I still have, somewhere in that office in all the boxes that people move for me, is a rope that makes a great demonstration of this. I unfortunately don't have that rope right now, so I'm looking for a good sort of cable-y thing. Actually, I don't even need help from somebody for this, as long as I can get this end out. Which I might not be able to do. Oh, there we go. Okay. So let's pretend that this is a piece of DNA, right? All right. It is linked on both ends. All right. It could be linked on both ends so it couldn't spin around like this. Okay. So if you feel free to stand up if you can see it, but it's linked on both ends. So it can't, when I turn it, it can't just spin around to release that stress. Does that make sense? Okay. So let's see what happens with this when I turn it. Here I can hold it higher, but this could be a circle and I could be doing the same thing. Okay. I have to be able to turn it and then link the ends, just like I'm going to do here, right? Otherwise, it would just spin and relieve it, right? Okay. So if I go ahead and I spin this and I spin it some more and I spin it some more and I keep doing that, it's getting really hard for me to spin it. This is why you need helicases. Or actually, which ones do you need? No, you need topopolymerases, sorry. So now, you see what it's doing? I'll hold this up. You see this? It did that on its own. That's supercoiling. I have introduced superhelical stress to it. All right? And if it could be, right now it's linked because I'm holding it. So that can happen with histones. See, here we got another one going. Okay? That happens with supercoils. So DNA does that too. And so when you have a plasmid is this big double-stranded. It's so big I'm not going to draw both strands. Imagine two strands, okay? Double-stranded piece of DNA. What happens is if it gets supercoiled, either over or under, and over or under is just whether you turn it left or right, it'll wind up like that. All right? And we refer to this as open circular. And usually when you see a piece of open circular DNA that you have isolated from an organism, so your DNA is supercoiled, bacterial DNA is supercoiled, any DNA we've grown up in bacteria or lab, right? And that's come out of that, so that's going to be supercoiled. But over time, what happens is it degrades a little bit and you get a nick in that DNA. And you nick one strand, two strands of DNA like this, right? If I nick one strand, now it could just spin as much as it wants to relieve all those supercoils. I can't make my arms go around and around and around. But essentially, if this had a lot of tension on it and I nicked it, it would spin around until all the tension came out. It was a nice big open circle like we see there. That sort of makes sense what's going on with that? Okay, so this one's supercoiled. And if I cut that linear piece of this piece of DNA, either one, and I make a double-stranded cut, so I can make a double-stranded cut, or if your DNA sits around long enough, it can actually get a nick and then another nick near enough that it turns into this. You could have linear DNA. So linear DNA would just be like this. It's just a straight strand. You've cut that strand twice in one spot, and so now it's open like that. Questions on those structures? This may feel like it's jumping around, but we're putting it all together for the stuff we need to know for lab. All right, so you can separate DNA on what's called an agarose gel. And what we'll do, we'll run gels tomorrow. We will put, we'll stain them. Unfortunately, the stain degrades quickly, so I have to come in early and pour the gels, or I'll pour them. If you're in the morning lab, what I'll probably do is I won't come in that early. You'll see me rushing around lab trying to get your gels ready, okay? But there will be time for that. So you can separate DNA in an agarose gel. What agarose is it's a polymer made from seaweed. It's a carbohydrate polymer made from seaweed. So a polysaccharide, carbohydrate. Okay, and essentially the strands act like, I'll say a sieve, and they slow bigger molecules down more than smaller molecules. So a sieve, a colander, is a sieve the wrong word? You could do a sifter. A sieve is correct. Okay. It's like scanning for gold. Yeah. Thank you. Act like a sieve, and should I just say colander? Like many colanders? It's actually like many colanders stacked. But also, I'm going to show you a video in a little animation in a second. Sieve, and slow down larger molecules more than smaller molecules. All right, so when we look at these structures here, in terms of larger and smaller, this is going to be largest. All these have the same number of base pairs. It's their shape. So this sweeps out more area and gets hung up more on the sieve than the other two. This is middle, even though it might not seem like it, and this is smallest. So the supercoiled one ends up getting scrunched up such that it can navigate through the sieve better than the linear, which must get hung up in places and then have to slide around like spaghetti. And then the largest one is this big circle. You could imagine trying to have spaghetti get through a colander if it was linked on the ends. Okay, let's go ahead and look at a video of this. So this is when we run agarose gels tomorrow. This is what's going to happen. It's going to look something like this. Sorry, I don't have a way to get you these videos. You can watch them in this thing I'm taping. But they're Flash videos, and Flash got outdated or outlawed or whatever a number of years ago. Okay, so the agarose gel looks like this. All right, it's like a piece of Jell-O, and what it has on the end are what are wells. They're little holes that you're going to be able to load your DNA samples into. And then what you do is you're going to hook them up to a power supply, where you have a positive electrode down here and a negative electrode up there. If I load my DNA up here, what charge does DNA have? It's negative, right? It's negative, one for every base pair, and the positive electrode's down here. So it's going to run this way, right? Whenever you do electrophoresis, just like your car battery, the positive will be red. So what one of my graduate school mates taught me was he said runs to red. So I just kept that forever. All right, so your DNA is going to run to the red. All right, so here we've got somebody pipetting their samples in that contain DNA, those wells, And I'll have to make some practice gels for you. Once again, DNA is negatively charged. And so if we turn on our power supply, what happens? There's your polymer mesh. And there's your DNA making its way through. And you see how the bigger piece is going slower than the smaller piece? Right? Okay? That's how that's going to work. I don't know if we have anything else afterwards. Let's see what else they did. They're maybe going to talk about how they're going to, oh, then they're showing you what this might look like. So if this was a restriction enzyme digest where they've cut with one and then cut with another and then cut with both at the same time, which is what you're going to do in lab, that's what the results would look like once we visualize them. And you'd have a molecular weight marker over here. So you'd have a bunch of pieces of DNA that you knew their sizes. So this might be 100, this might be 2, 3, 4, 5, 6, 7, etc. 100 base pairs long. And we would use those to line up. So if it was actually true that I was going 100, 200, 300, 400, 500, 600, 700, that's 700 long, right? This one, they made it so all of theirs lined up exactly. If I had something in between, I'd have to, 1, 2, 3, 4, 5, 6. If something was here, I'd say it's 550, right? I just kind of make a guess as to what I thought it was. An educated guess. Oh, look at that. That's a better photo. All right. Questions on that, how that works? All right, we're going to be doing that. Now, why am I telling you about this supercoiled linear and open circular? Well, when you cut, if you're cutting a piece of DNA, right? I've got a piece of DNA here. Let me do it here. Wow, okay. Maybe if I do this, I'll get it back. Aha, all right, great. So if I cut a piece of DNA, let's say, I've got a circular piece of DNA, or I've got a supercoiled piece of DNA, and I cut one of these. I cut it there. What am I going to get out of that? I'm going to get a linear piece of DNA, right? What if I cut this circular piece of DNA there? I'm going to get a linear piece of DNA. I'm going to get either of those, right? If they're the same piece of DNA, they'll be the same length. Okay? So either way, I'm going to get this linear piece of DNA, and linear pieces of DNA are going to run according to their size. So when we looked at that thing with the molecular weight marker, and we'll have another one later to look at, with the molecular weight marker, that molecular weight marker is all linear, and everything I run is linear, then I'm going to be able to match the sizes. But if some of my stuff's supercoiled and some of my stuff's open circular, that's not going to run, I'm not going to be able to call sizes off of that, off of a linear marker, right? Because it's different shape, it runs differently. That makes sense? Okay, so the goal when you do your experiment, you're going to cut pieces of DNA, and the goal is that you will completely cut the piece of DNA. So if you have this or if you have this, you will completely cut it, and you won't have to worry about any supercoiled or open circular DNA left. But if it doesn't completely cut, it's going to give you some different things to look at. So let's see, I didn't grab a picture of supercoiled DNA, but let's go supercoiled DNA gel. Perfect. This one. This is kind of the sort of thing you'll see. Oh my, okay, now it's not doing it again. All right, here we go. So when you run a gel, this is the sort of thing you're going to see. This should say linear, but so what you'd end up with is uncut DNA might look something like this. I don't know really, I didn't do this lab last year, so I don't have a good feel for what the quality of your DNA is. It was bought in 2022. It could be all nice and super coiled. It's probably not going to be this bad, all right? But it may have some nicked in it. So uncut DNA will look like this, right? And you will run a control where you don't add any restriction enzymes, you don't cut any DNA. And so this is on there to tell you, this, you don't use this for anything, right? You see a band for that, it doesn't count, except for the linear, right? If you cut something once, it would look like this. Does that make sense? But this is biggest. This nicked circles is the open circular. It sweeps out a big space. Linear, not so much space, and then supercoil is nice and compact and it goes really fast. Yes?
So what's the difference between cutting it once and cutting it twice? Like,
Well, nicking it once, or are we talking about nicking it, or are we talking about
Yes.
cutting it? Okay. Nicking it is cutting one strand. So DNA is double stranded. You nick it, you still have if you have a circle and you nick it, you still have a circle.
okay.
But this can spin around now and relieve the supercoil stress. You cut it again, if you nicked it on the other side close, then these will come apart and become linear.
And linear is different than the like numbers on the other side?
This linear is the same as you
Oh,
could
it
compare.
is. Okay,
You could
okay.
compare this linear with these.
Okay,
linears.
okay.
So whatever this plasmid is, it's much bigger than all of these pieces they made from, which you'd expect, right? If this is let's say this represents all the pieces of that plasmid, right? Which one would guess, if let's say it's one, well, it doesn't really matter how many restriction enzymes it is. Sometimes you can have some stuff that runs off the bottom. So if you have stuff that's really small, it can run off the bottom. It also doesn't stain well. But let's say that's all the pieces of our plasmid. How big is that plasmid? What was that? It's greater than 2319. I can actually be, can anybody think about how we'd be more precise? You could just tell me how. You don't have to do it.
Add them together.
Add them together, right? Whatever that is, I cut it with 1 or 2 or 5 restriction enzymes. I ran it down on the gel and I got a bunch of pieces. And as long as I see all the pieces, you add them up, that's how big the plasmid is. Okay? Questions? All right, so I've been talking about this concept of restriction enzymes. What restriction enzymes are they are proteins that will bind to specific sites on DNA and they will cut it, okay? When I say cut it, they're going to cut the backbone, they're going to break a bond on each side. So a restriction enzyme is going to break a bond at one site on the backbone and then across on the other strand, pretty close, another site on the backbone, all right? How's it going to break the bonds? What reaction are we probably using because this is
Hydrolysis.
Hydrolysis, right. We're going to add water to break that bond. Okay? So an animation of what that would look like is this. This isn't very exciting, but. So this is some restriction enzyme, EcoR1. It's named after E. coli. It's R1. These restriction enzymes are defense mechanisms that these bacteria use against viruses. Okay, and what we've done is we've found all sorts of things they do. We co-opted it to be able to cut things we want to do molecular biology. So that's the recognition sequence for EcoR1. Let's go back. All right, so it lit it up, and it was between here and here. So EcoR1 only cuts at this 6 base pair sequence. It needs to have an GAATTC. Needs to recognize the right functional groups in either the major or minor grooves for that, and then it will bind here, and it will hydrolyze the backbone in one place and in another place. I think I got those right, but I may not have. So here it's coming over. It's not going to show us adding water, but it's going to cut. So it recognized GAATTC, and then it cut right after the G, and then on the other strand right after the G. So imagine this is 5' to 3', and then this one's going to be 5' to 3'. So it's a symmetric sort of thing. Does that make sense what it's doing? Now, because we want to learn stuff, we're not going to use this tomorrow, but you can take, if you have these things, you got sticky ends. What they mean by sticky ends is that this A can match with that T, this A can match with that T, this T with that A, this T with that A. 2 hydrogen bonds each. And so we could actually use that, or we can actually bring any 2 DNA strands back together. But if we specifically wanted this end to match up with another end, we'd have to have the same sticky ends. And so we'd use an enzyme called DNA ligase. So it's going to have that phosphate. And there we have, what reaction was that ligase was catalyzing? Condensation, right? We eliminated water. Questions? Okay. All right, so then you can get, there are all sorts of restriction enzymes. You can buy them. Ignore the average cleave size, but I want you to look at some restriction enzyme names and where they cut. I realize that's going to be hard for you to see in the back, so I'm going to make it super big. You're going to be using EcoR1 and PST1 tomorrow. All right, so we just talked about EcoR1. It has this sequence G. Oh, that's Biochemistry. EcoR1 right there has a sequence GAATTC, and what this is showing you is that on the top strand, and whenever you see 2 strands written and nobody tells you which is the 5' end and which is the 3' end, it means that this is the 5' end and this is the 3' end, and the 5' and the 3'. Let me, I'll just write it on this one. So this is 5' to 3', and this is 5' to 3'. And I hope you're getting the feeling that the directions matter. What direction the polymer goes matters in these things. For nucleic acids, well, for everything. For that matter for proteins as well. So it's going to cut here and here. That's how you read this chart. BAMH1 is going to cut. It recognizes a different 6 base pair sequence, but it's going to cut kind of the same place, right? After the first G. Let's see, do we have PST1 on this chart? There it is. So here's the other one. And this one's going to give you kind of different ends. You see how it cuts down there? Instead of cutting here and here, it cuts there and there. Okay? And so different sequence again, and then now it's going to cut right before the last G in both cases. We've got a lot of cutting at G's, but it's not always the case. If you look at TAC here, it doesn't. Here's an interesting one. Okay, so we have these sticky ends, right, that can come back together. What about this one? What kind of ends would we get? If we cut that, we're going to end up with G, 3', C, 5', C, 5', G, 3', like that. And then the sequence will go on. It'll have some sequence. This is just whatever sequence is there that they would give us. So a bunch of DNA sequence, GGCC, it will cut like that, and then it'll give you CC and a bunch more sequence. Does that make sense what's going on there? Okay. That's how restriction enzymes work. So we've talked about restriction enzymes, and we've talked about ligase, right? Let me go ahead and just define those here. So restriction enzymes are usually proteins that bind to DNA at specific sequences and cleave it. Okay? All right, yes.
For that top part, if you scroll up a little bit,
Yeah, how do I know what's 3' and 5'? Whenever anything's written and they don't indicate it, that it's going, the top is going to go 5' to 3'. It's just convention. It will always be this. The top will go 5' to 3', and the bottom will go 5', 3'. So it cuts here, right? So this is going 5' to 3'. And so that has to be 3'. Yeah, no, it's a great question. Remember, if you ever have a question, somebody else does too, right? At least two other people in the room do too. The same question. Other questions? Yeah.
You said we were using 2 restrictive enzymes.
Yes.
The Eco R1. What was the other one?
PST1. And I'll tell you tomorrow, but it's this one. So there you can see what the sequence is. Okay. So let's go ahead and do a plasmid map. Oh, actually, let's not do it first. Let's actually first define ligase. Ligase is an enzyme that will join two pieces of DNA. It will only do so if the pieces of DNA have complementary ends or they have blunt ends. We'll go up and we'll talk about what blunt is. Let's say complementary sticky ends. Comma, or blunt ends. Okay? Let's talk about what that means just a little more detail. These are blunt. All right, do you see how if ligase came in, it doesn't have any problem linking the two? Okay, it just can take any two blunt ends and link them together. However, let's say I had I'm going to scoot down just a little bit. I'm going to leave us our restriction enzyme sites up here. Let's say I had a PST1 end. So the PST at one end is going to go like this. It's going to go C, and it's going to be a 3' there, and it's going to go, oh no, it's going to go, sorry, it's going to go C, T, G, C, A, and then I'm going to have a 3' end. And on the bottom strand, it's just going to go up to G is where it was cut. And that's my 5' end. Okay? And let's say I wanted to link it with one of this TAC1 enzyme. And so that's going to have an end that's going to look like this. It's going to go 5' C, G, A, and it's going to go 3' T. Can those link up together? Are they complementary? They can't because they're missing, right? It's really bad, actually. The sequences don't match. This one's 4 longer than that one. I can't just flip one of these over because then I need to link a 3' to a 5'. I can't flip it and then link a 3' to a 3'. The directions are wrong. So these do not. Okay, so cannot ligate. Now we could modify these ends, and we may talk about that later, but not today. But you can't ligate those two together. The ends are not complementary. Questions? Bless you. All right, let's do a practice problem here. Let's do a restriction enzyme map. I haven't even talked how to do it, so I'm going to demonstrate how you do one. Okay. All right, so this company, Carolina, has these things. These are great. You guys may not be able to see it out well in the back, and I apologize for that. If you want, there's a seat here, but I'm not going to be able to fix it very much. I can actually draw these things bigger. Let me label it larger. And actually, it's so small on my screen. Let me get my glasses out. Okay, so what we have up here is I've got some enzyme cuts going on here. So I've got a BAMH1 enzyme. I'll just use a big B for that. I've got an EcoR1 enzyme in this lane that we have the products from. And then I have both. All right, hopefully that's clear to people in the back. And then the sizes here, I'm going to change these numbers because they talk about it being number of base pairs, but it's 20. You never do this on something that's 20. I'm going to add 2 zeros to it. So we're going to pretend this is 2,000 base pairs. And the way you'd get these sizes is you'd compare them to a molecular weight marker that they haven't included in these problems. But you will have a molecular weight marker in lab. This will be 1,100 base pairs. This will be 700 base pairs. This will be 600 base pairs. This will be 400 base pairs. This will be 300 base pairs. Okay? Can everybody see that? All right, so we've got a plasmid. We cut that plasmid. And so this is the way I would figure out what the restriction map is. Okay? I would take, I've got some plasmid. I don't even know what size it is yet, right? But I'm going to make a map for each single cutter, and then I'm going to make a map for both. And I have to put, and all of them have to be consistent with all the data. Okay? So I'm going to start out with my BAMH1. All right? I cut it, and I get one piece. Okay? How many times did it get cut if I got one piece? Once. So I'm going to make a place for a cut right there. I'm going to label that cut at 0 in terms of a map. I could label it anywhere. I could call it 100, 400, 800, whatever. One cut anywhere is going to give me one piece. And how long is it?
2,000.
That's 2,000 base pairs long. So how big is my plasmid? 2,000. Okay? So this is 2,000 base pairs according to this. So hopefully, when I do my next cut and add up all the pieces, I'll also get 2,000, right? Okay? So next cut is with EcoR1, I think. Yes. EcoR1 cuts 3 times, right? All right, what do we get from that? We get a 1,100, right? We get a 600, and we get a 300. And I can look at this one right now and say, hey, my life isn't even very complicated. There's some things that can happen to complicate this, like bands can lie on top of each other. So then you'd have I could have 2 600s, I wouldn't be able to tell. And so I'd have to figure that out based on all the other cuts. I have to reason my way through that. This one looks like it's simple. So I'm going to have it cut, okay? I'm not going to worry about this map relative to this map yet. I'll worry about the two of them together in my together map. Does that make sense? So I'm just going to make this one not worrying about the other map, pretending the other one doesn't exist. So what I'm going to do is I'm going to say, well, first of all, I've got 3 pieces. Yeah, how many times does it get cut? 3 times. Okay, my first time, I'm going to put it 0. Actually, how big is it? 2. It's still 2,000, right? All right, great.
Are they always going to be?
They have to be the same. It's the same plasma. They have to be the same. So if they're not, that means you have to do some more reasoning to figure out what's going on. Like bands on top of each other.
So
Or
it's different enzymes, but
it's different enzymes, it's cutting the same plasmid. We're making a map of the same plasmid. Everybody with me?
Yeah.
Okay. So we cut what? So I'm just going to call that 0 because it's easy for me. How far away from that one might I cut? How far away could the next cut happen that would be consistent? Halfway down? Okay, specific, 1100, right? If I cut at 0, we're going to call my next cut at 1100. Okay, great. So we took care of, so that means this fragment right here is 1100 base pairs in length. Where would my next cut be? 1700 is a great choice, right? Because I have a 600. So if I make it cut at 1700, that would reflect a piece that's 600 base pairs, right? Okay. And then you don't really need to put the base pairs on the locations on the plasmid map. And then what's left over? 300, right? It's already there. I will leave them on the fragment side. Okay, you guys with me for this? All right, note that the plasmid map itself is not an 1100, a 600, and a 300. It is a cut at 0, a cut at 1100, and a cut at 1700. Okay, and I know these cuts are eco R1 because I put eco R1 in the middle. I don't have to label each cut. I could. Same thing up here. All right, but these are like addresses of where the cuts happen. Okay, the address of where I live is, Stone Mill Drive. Okay, is where I live in Salem, and that's 6 miles from work. Let's say, actually, it's 3.5 miles from work. Okay, 3.5 is not my address. Stone Mill Drive is my address. Does that make sense? I want addresses, not distances, for these maps. Jacob.
So I'm curious, tell us, how do we know it's in that specific arrangement and it's not like first cut at 600 and then it cut
Right. Okay, good. Great question. So how do we know that it's 1100, 0, 1100, 1700, right? As opposed to going and putting the 600 and then the 300 and then the 1700, right? There are multiple arrangements here that would work. We don't know at this point, all right? With 2 restriction enzymes, we're going to bring them together, and that's going to, it's not actually 2 restriction enzymes is not going to invalidate any of this. If we had 3 restriction enzymes, suddenly we'd have to get that order correct. All right, and I'm not going to give you so you'd have to reason more to get that order correct. I will not give you anything over 2 restriction enzymes. Does that make sense? But if you add more and more and more restriction enzymes, suddenly not all these possibilities work. Yes.
So the plastic map is the draw line from the 0 to the 1100 to the 1700.
The plasmid map is what I've left.
Okay.
That's your plasmid map. So it's 3. You have 3 cuts, and you've given me 3 locations for those cuts, not the distances between them. You can write the distances on it to help you figure it out, but if you don't give me where they cut, you're not going to get much credit for it. Okay?
And then the both, does it even matter that it's there? Or?
Oh, it matters a lot. It matters a lot. We have to put it all together.
Oh, okay.
Okay? So I'm going to erase these to make room for my both. All right, we know both is going to be 2,000 base pairs at this point, right? So let's see if there's any funny business. I'm going to pull the both numbers over to here so we don't have them in the way. So we've got a 700, right, for both. 700 base pairs. We've got a 600 base pairs. We have a 400 base pairs. And we have a 300 base pairs. All right, does that add up to 2,000? Great, no funny business. But now I have to make this work. I have to make both maps work, right? I have to essentially take one map and superimpose it on the other. The easiest way I find to do it is I take the more complicated map and I keep it the way it is. And then I rotate the other map to make it give me the right fragments. Okay? So let's go ahead and let's take the more complicated map and just duplicate it. 0, 1700. And by the way, with what Jacob said, if you start doing 3 restriction enzymes or more, this method might not work. You might have to be, there might be more reasoning involved. These are, what enzyme was cutting at these spots? EcoR1. So I'm going to put an E there, an E there, and an E there, so I know when I grade it, I do what I was doing. All right? Otherwise, you will not get much credit if you just show all the stuff and don't tell me which enzyme's which. So EcoR1 cuts at all those places. Now I need to look at this. What in my EcoR1 cut here, which band went away? Which band or bands? The 1100. Okay, did we get new bands? We got a 700 and a 400, right? Do they add up to 1100? That means that the BAM H1 must be cutting in this piece, right? How far into that piece would it cut? Either 700 or 400, right? So I could go ahead and I could say at 400, then I'm going to end up with a BAM H1 cut. Okay? I could also have said at 700, I've got a BAM H1 cut. I wouldn't say both. But both would be consistent because both would give me this gives me a 400 piece and a 700 piece. This cut gives me a 700 piece and a 400 piece. They're both consistent with the data that I have. If I added a third restriction enzyme, one of those probably wouldn't be true anymore. One of those probably wouldn't be good. Like I said, we're not going to go to that level of complexity. And I'm going to erase that so that it's clear that you want to give me one of those, not both. Questions on how I did that?
So just choose one?
Choose one, yes. Whichever one, you need to choose something. Whatever you choose needs to be consistent with all the data. So is this consistent with all the data? So these maps, right, were consistent with their single cuts. All right. And this map, is it consistent with the single cuts? Well, if I just use EcoR1, I get an 1100, a 600, and a 300, right? And I get an 1100, a 600, and a 300. It's consistent with that. How about with the BAM H1 cut? Well, if BAM H1 cuts once, if there's no EcoR1, I get one big piece that is 2000, and that's what I get. And it's consistent with the double cut as well. It needs to be consistent with all the data. Sophia, do you still have your question, or did I get it?
No.
Okay. Questions? All right, well, why don't we go ahead and spend the last, I don't know, is it 6 minutes? Go ahead and start working on your pre-lab. The pre-lab, if you look at it, I think it says at the top, does it? Where does it, or is it at the top of the questions? Page back to the questions if it doesn't tell you which you have to work. There's only 4, is it 4 or 5? There's only 4 of these you have to do. So if after this lecture you've been able to do all of them, great. If you can't do all of them after the lecture, I'm available 1 to 5 today, but also this thing will work you through a set of other questions that will help you learn how to do it. And if you have questions right the next 6 minutes, I'm happy to come over and help out. I'm going to cut the video though.